Okay, thank you so much for the invitation to speak for the CFAR, and thank you for the introduction, Pavitra. So I'm going to tell you about our studies on HIV expression, and I'm going to start sort of with our early studies where we first started to study the expression of proviruses in single cells. And then I'm going to tell you, and some of those studies are already published, but I'm going to just give you that for background. And then I'm going to tell you about our new studies where we're looking at HIV expression in single activated cells and resting cells in donors on long-term antiretroviral therapy. So before I do, I want to introduce my lab because, of course, they're the ones who have put so much effort into the studies I'm going to tell you about in this talk. So most of the studies I'm going to tell you about were done using a technology that was developed by Ann Wigand in my lab and John Spindler. These are both biologists in the lab that developed this amazing technology where we can isolate single infected cells and we can look at the expression of proviruses in these single cells that are collected from donors. In the cell subsets, those studies were done by Andrew Music, who was a master's student in the lab. And also he looked at the fraction of cells within expanded cell clones that have HIV RNA, and I'm going to tell you about those studies today. And then in the activated and resting cells, those studies were done by Jen Grosvenor, a postdoc in the lab, and Rachel Slukas, who's a master's student in the lab. So I'm just going to give you this for context. I know you all know what antiretroviral therapy does to HIV, but just for context to get you oriented for what I'm going to talk about today. So as you know, when individuals initiate ART, their levels of viremia decline typically about 3 to 4 logs. And so what was hoped when ART was first developed, combination antiretroviral therapy was first developed, is that these levels of viremia would continue to decline until they went away completely and it would cure HIV infection, which of course, as you all know, did not happen. But instead what happened is these very low levels of persistent viremia were maintained during therapy. And these levels are typically at about a single virus particle per mil of plasma. So we and others asked, where is this persistent viremia coming from? So we looked first at the number of infected cells before ART and the number of infected cells during ART. And what we found is that although the level of viremia declines 4 logs, the number of infected cells only declines by about a log during antiretroviral therapy. So many of these infected cells are persisting during ART. And they must be persisting in a latent state because the levels of viremia are very, very low during treatment. So we were interested in characterizing the proviruses in these cells that persist during ART and also looking at their levels of expression, because although most of these cells must be latently infected, some obviously have proviruses that are being actively transcribed and generating the virus particles that we can measure and sequence in the plasma of individuals during therapy. So we wanted to understand what fraction of the cells have proviruses that are being actively transcribed during therapy and what fraction of the cells have proviruses that are completely quiescent. And so, as you know, before initiating antiretroviral therapy, there are ongoing cycles of viral replication with new cells infected every day. As the virus replicates, it accumulates genetic mutations. And by the time an individual has been infected for about 3 years or so, if you do single genome sequencing on the RNA genomes in individual virus particles in the plasma, what you find is that almost every single RNA genome is unique. So if you sample about 50 different proviruses from the plasma of an individual who's not on treatment, you'll get 50 different RNA sequences. So the genetic diversity of HIV is very, very high, as I'm sure you all know, but we exploited that genetic diversity to be able to develop the technology that we did where we can sequence single RNA molecules within cells that persist during therapy. And I'll tell you a little bit more about that in a moment. So although it's been known for years that there's ongoing cycles of viral replication and new cells infected every day without treatment, what we didn't know, but we discovered in 2014 in the HIV Dynamics and Replication program and in other labs around around the country and the world, including at the University of Washington, is that some of these cells actually persist despite being infected. And when they persist, these T cells can then undergo clonal expansion or cellular proliferation, resulting in these populations of cells with identical proviruses, most of which are probably latently infected, but some of which have proviruses that are transcriptionally active. And if the provirus in these expanded cell clones is intact, then they will also generate virus particles. So before initiating therapy, there are two sources of viremia. One is the ongoing cycles of viral replication in the new cells that are infected every day. And the other are these cells that were infected days ago, weeks ago, months ago, even years ago, that exist in these populations of clonally expanded cells, some of which have proviruses that are being transcribed and generating virus particles that are also contributing to viremia before ART. So when ART is initiated, what we know now is that the ongoing cycles of viral replication are virtually halted. And what you're left then with is these populations of clonally expanded cells. These cells were infected before ART, many days before, months, even years before. They exist in these populations of clonally expanded cells. And if they have intact proviruses, they can be expressed, they can generate virus particles, and that's this low-level persistent viremia that we see in individuals on ART. So there have been several reports now that have shown that these expanded clones can have intact proviruses. They can generate virus particles that are replication competent. And these populations of expanded cell clones with intact proviruses are now called repliclones. And that's a term that was coined by Lou Halvis et al. In their recent paper, actually in our recent papers, I'm a co-author on that, in JCI in 2020. So we set out to ask, within these expanded cell clones, what fraction of the cells within a single clone are latently infected, and what fraction have proviruses that are being actively transcribed? And we actually published that result in 2019. So I'm going to jump right to the punchline, but I am going to just show you some of the data that we have that shows that, you know, where these results come from. So it turns out that it's a median of about 5% of cells within expanded cell clones that have proviruses that are transcriptionally active. So about 95% of the cells within these expanded cell clones are latently infected. However, there's a wide range. So some expanded cell clones only have about 1% of the proviruses that are transcriptionally active, while other expanded cell clones, it's up to about 65% at a given point in time that have the proviruses that are transcriptionally active. So it's actually a very wide range. So what makes a provirus within a clone reactivate and start generating unspliced RNA and virus particles is not yet known, but that's a major focus of our research and something that I'm going to address in this talk today. Okay, so how do we do this? If we're going to ask what fraction of the cells within an expanded cell clone has proviruses that are transcriptionally active, and what fraction has proviruses that are quiescent, we have to isolate single infected cells from within a clone. So this sort of drawing here is supposed to represent T cells from within a single expanded cell clone. They all have identical proviruses because they resulted from the proliferation of a single infected cell. And so we have an assay where we can isolate these cells. We can confirm that they're from a single clone by sequencing the proviruses. And then we can assay the single cells for the presence of unspliced HIV RNA. And when we do this, we find that a small fraction of the cells have unspliced HIV RNA. And if this provirus is intact, then it's these cells that are producing virus particles that result in this low-level persistent viremia during ART. And so from this assay, we can count the number of cells that have unspliced RNA, the numbers of cells where there's no detectable unspliced RNA, and then we can sort of do the simple math and show that in this hypothetical example, 25% of the cells have proviruses that are being actively transcribed, and 75% of the cells are latently infected. And so we set out to do this on a number of different expanded clones. So technically, the way that this is done is we take the cells from an individual. It can be PBMCs, it can be lymph node mononuclear cells if you're interested in looking in the tissues. But we take the infected cells and we spread them across multiple aliquots. We then extract the RNA, synthesize the cDNA. We spread the cDNA across multiple 96-well plates so that we can amplify single molecules of cDNA. We then sequence those molecules and we make phylogenetic trees. So when we see different RNA sequences, then those are RNA molecules from different infected cells, because we know from previous studies that about 90% of infected cells have a single provirus. But when we see identical RNA sequences and they are from the same aliquot, so for example, on this phylogenetic tree, we have 5 identical RNA molecules. They're all from the same aliquot of infected cells, and we have very few infected cells in each of these aliquots. So we can assume that these RNA sequences resulted from a single infected cell that had 5 molecules of unspliced HIV RNA. And so by doing this, then we can calculate, and if we see identical RNA sequences from different aliquots, then we can assume that there were different cells with the same provirus, probably an expanded cell clone, where multiple members from that clone had proviruses that were being actively transcribed. So from this data, we can calculate back to the number of infected cells that we had. So for example, if in this red aliquot, there were 100 infected cells, let's say, we found only 2 different RNA sequences. We may have found multiple molecules of those, but only 2 different sequences. Then that would be from 2 different infected cells. So we would say then that 2 of the 100 infected cells in this aliquot had HIV RNA, and therefore 2% of the infected cells in this aliquot had proviruses that were being transcriptionally active. And so using this method, we can then sort of take the average across all of the aliquots of the number of infected cells in the aliquots, the number of those cells that had HIV RNA, and then we can come up with a sort of an average for this sample. So in this hypothetical example, it's 3% of all of the infected cells that had HIV RNA. And so using this technology, then we first just looked in untreated individuals. So in individual, this is just showing one example. We looked at about 10 different patients who were in chronic infection, and they were not on ART. And we just performed this CARD SGS assay, which is the assay that I just described to you. We call it CARD SGS, cell-associated RNA and DNA single genome sequencing assay. It's a mouthful, but we just call it CARD SGS. And so when we do this in individuals who are not on ART, we do it exactly the same way. So we make all of these aliquots with very few infected cells in each of these aliquots. We then assay them for unspliced HIV RNA. And in untreated individuals, we find these cells that have very high numbers of RNA molecules. So when you see these squares, each of these squares represents a single RNA molecule, unspliced HIV RNA molecule. And when those squares are the same color, they came from the same aliquot. So for example, all of these orange squares here are unspliced RNA molecules from a single infected cell within this one aliquot. So in untreated individuals, we see these single cells that have very high numbers of unspliced HIV RNA. And by the way, the reason we focused on unspliced is because obviously when HIV is expressed, you have lots of multi-spliced transcripts, but when you have enough of that TAT produced, that's when you start making these unspliced RNA molecules. And it's those that are required to generate virus particles. And so we think that looking at unspliced RNA really gives us a really good look at those proviruses that are really being transcribed at high levels. Anyway, so in these individuals who are not on treatment, we see these single cells with very high numbers of unspliced RNA molecules, and we call these high expressing cells. And it's these cells that are probably contributing to the very high levels of plasma viremia that individuals have before they initiate ART. So we then did this same assay on individuals on ART. So this is just showing one example, and this is an individual who has had their viremia suppressed on ART for 9 years. And when we perform this CART-SGS assay, this tree looks very different from the one that I just showed you from an untreated individual. In this individual, we don't find any of these highly expressed cells, and this is consistent with all of the donors that we've looked at whose viremia is suppressed on ART. So instead of these high expressing cells, what we see instead are these populations of identical RNA sequences, but not all from the same aliquot, but across multiple aliquots. And so I'll just blow this up for you. And so what we think that this means is that you have infected cells in different aliquots, all with identical proviruses, some of which are transcriptionally active and making unspliced HIV RNA. So these are the expanded clones that are revealed with ART. So these expanded clones, they exist in pre-therapy, but when an individual goes on ART, many of these infected cells die, and you're left with these expanded cell clones, as I described earlier. So with ART, it reveals these expanded cell clones that were already there before, but these high expressing cells are now gone. And instead, what we're left with, those high expressing cells probably all died or in the early months or years of ART. So by the time an individual has been on ART for 9 years, you're left with these clonally expanded cells with some members that have proviruses that are transcriptionally active. And of course, we can determine if these clones are have replication competent proviruses by using the viral outgrowth assay and matching the virus that grows out to these expanded cell clones. And that's how we identify these expanded cell clones that have replication competent proviruses within them. So the unanswered questions were, what fraction of proviruses within expanded clones express HIV RNA? I already told you the answer, but I'm going to show you a little bit of data just so you can see how we got that answer. But the answer is that it's an average of 5%. So within these different populations of expanded cell clones, about 5% of the cells within each of the different clones have proviruses that are transcriptionally active, while about 95% are latent. So keep in mind that these expanded cell clones have identical integration sites. So it suggests that the integration site may not be what is important when it comes to determining whether that provirus is transcriptionally active or silent. Because within the same clones, we see some of the cells that have proviruses that are transcriptionally active and other cells that have proviruses that are quiescent. So then we wanted to know if this fraction was different for clones that have replication competent proviruses versus clones that have defective proviruses. And which T cell subsets harbor and express proviruses in expanded cell clones. So if it's not the integration site that is important in determining whether a provirus is expressed or not, then is it the state of the cell? So in other words, if we have an expanded cell clone where some of the members have proviruses that are transcriptionally active, others of the cells within that same clone have proviruses that are quiescent, are the cells within that clone in different states? So in other words, are some of the cells within a single clone activated while others are resting? Are some maybe effector memory cells while other are central memory cells? And so maybe it's the state of the cell that determines whether or not the provirus is reactivated or not. And so we wanted to know the answer to that question. So I'm just going to show you some of the data where we determined that it's only about 5% of cells within expanded cell clones that have proviruses that are transcriptionally active. So this study was done by Andrew Musick as a part of his master's thesis, and he actually published these results in 2019. So he looked first in 3 expanded cell clones that had intact replication competent proviruses. And within these clones, he assayed single cells for the presence of unspliced HIV RNA. And the different sizes of these pies reflect the different sizes of these clones within the individual. So this AMB1 clone was a very, very large clone. He actually assayed 1,200 single cells from within this specific clone, and 2% of them contained unspliced HIV RNA. He then looked at 2 other clones that had replication competent proviruses. In one of them, 0.8% of those cells had unspliced HIV RNA, and in the other 5.5%. So it was a median of 3% of cells within expanded cell clones that have replication competent proviruses that have where those proviruses are transcriptionally active. He then compared this to clones that had defective proviruses, and the median was 5%. So there doesn't appear, there's no apparent difference. So if a clone has an intact proviruses versus a defective provirus, it still seems to be right around 5% of the cells within these clones where the proviruses are transcriptionally active versus latently infected. Those were the results that we already published. And I'm going to now tell you about the studies that we're doing that we haven't yet published. So as I already said, as I already told you, the integration site, although there are some studies that claim that the integration site is what is important in determining if a provirus is transcriptionally active or not, and that may be true, but when you look at the clones that persist on long-term ART, you may have already selected for a population or populations of expanded cell clones that have proviruses that are just not readily reactivated independent of the integration site. So we may be looking at specific clones here that persist on long-term ART, right? Versus those that might be there in short-term ART where the integration site might matter. But in this AMB1 clone, we had already published that 90%, 98% of the cells within this clone were latently infected. Only about 2% had proviruses that were transcriptionally active. This AMB1 clone was the sole source of persistent viremia in this individual. So this 2% of cells must have been producing all of the persistent viremia that we were detecting in this individual. Of course, we didn't look in, we haven't assayed, all of the tissues in this individual. So it could be that in some tissues, it could be a much higher fraction of the cells that have proviruses that are transcriptionally active. In this case, we looked only in the PBMCs. But so we asked if this 2% of cells that have proviruses that are transcriptionally active within this clone, could they be activated cells? Francesco Simonetti, who characterized the provirus within this expanded cell clone, he had published and suggested that this clone was responding to a cognate antigen, which was probably a tumor within this patient. So could this 2% of cells within this clone be the activated cells? And could those activated cells have proviruses that are transcriptionally active, while the resting cells maybe had proviruses that were transcriptionally silent? So in other words, do activated cells within the same clone, cells that have the HLA-DR surface marker, for example, do they have proviruses that are transcriptionally active? And do the resting cells have proviruses that are transcriptionally silent within that same clone? So we set out to answer this question. And the reason that we thought that this might be the case is because resting CD4 positive T cells are thought to have limited or inefficient proviral transcription. We know that when we take these cells and stimulate them ex vivo, and when we stimulate them ex vivo, we really hit them hard with anti-CD3, CD28. And when you do that ex vivo, you can sometimes reactivate these proviruses and get them to generate virus particles. And that's exactly how the viral outgrowth assay works. So we thought maybe that's exactly what's happening in vivo. Maybe the resting cells have transcriptionally silent proviruses, and maybe the activated cells have transcriptionally active proviruses. And so when you look within an expanded cell clone and you see some small fraction of the cells that have proviruses that are transcriptionally active, maybe those are the activated cells. And so the first thing that we did was we just collected samples from 8 participants where we could just separate out the activated cells from the resting cells and just look at the overall population. Do we find higher levels of HIV RNA in the activated cells in vivo than we do in the resting cells in vivo? And so these 8 participants came from several different studies, ACTG studies, NIH studies, and the SCOPE cohort. And what I want, the demographics are in this box here on the left, but what I want to point out is that these individuals were on ART for many years. So they had their viremia suppressed for many years, an average of 9 years. So we're looking here only at individuals on long-term ART. And I think that's really important because on long-term ART, we may have selected for a population of cells where the proviruses are very resistant to reactivation. So in other words, in the early months or years of ART, all of those cells that had proviruses that are easily reactivated may have already died off, right? And so we're looking at the population of these infected cells that really persist on long-term ART, that we want to try to target and kill these cells to cure HIV infection. So from these participants, we collected the PBMCs and sorted them into HLA-DR positive and HLA-DR minus subsets. So we call these resting and activated cells, but of course there are many markers for activated cells that are not accounted for here. And the reason we did this is because when we tried to sort using multiple markers on the surface, activation markers, we really couldn't get clear separation of populations. So we decided to sort them based on HLA-DR positive, DR minus, where we could get very clear, distinct populations, and then to characterize the cells with RNA-seq. And that's what we're doing right now. So from these DR positive, DR minus cells, we used our CARD-SGS assay, and we asked what fraction of the activated cells have proviruses that are transcriptionally active, and what fraction of the resting cells have proviruses that are transcriptionally active. And as you can imagine, we expected that in the resting cells, we would find that the proviruses, virtually all the proviruses are latent. And in the activated cells, that most or many of the proviruses would be actively transcribed. So activated proviruses, just like the activated cells. That's the hypothesis. And just as a reminder, the CARD-SGS assay, we can detect a single cell with HIV RNA and then calculate the fraction of the infected cells that have HIV RNA. And we can also calculate the levels of RNA expression in the single cells. So I'll show you that as well. So we first looked in the resting cells in these 8 individuals. And so what you're seeing on this graph is on the y-axis is the estimated percent of the infected cells with HIV RNA. So these are resting cells. So we expected that in these 8 different individuals that we would find virtually no resting cells that had HIV RNA. But instead, what we found here is that it was about 1 to 8% of the resting cells that had proviruses that were transcriptionally active and had unspliced HIV RNA, suggesting that if they're intact, they can contribute to persistent viremia on ART. So this was a surprise to us that resting cells actually can have proviruses that are actively being transcribed. So then we looked in the activated cells, and we expected to find that most of the infected activated cells would have transcriptionally active proviruses. But to our surprise, we actually found that the activated cells did not have any higher proportion of proviruses that were transcriptionally active. In fact, most of the activated cells had proviruses that were completely quiescent. And there was no difference. So 4% of the activated cells have proviruses that are being actively transcribed, and 5% of the resting cells have proviruses that are being actively transcribed. And again, this is on long-term ART. So it appears that activated cells, so cells that are likely activated by their cognate antigen, undergo massive clonal proliferation, but that is not enough in vivo to reactivate these proviruses from within them. So what we concluded is that greater than 90% of both resting and activated cells that persist on long-term ART have quiescent proviruses at a given point in time. So then we looked at the levels because we thought, okay, maybe the fraction of activated cells that have HIV RNA is the same as resting, but maybe their levels are much, much higher. And so we looked at the levels of HIV RNA expression in these resting and activated cells that had transcriptionally active proviruses. And we actually found most of them to have very, very low levels of RNA. Most of these cells had just a single molecule of unspliced RNA at a given point in time. Some of them had a few molecules, 2, 3, 4, or 5 or so molecules, but whether the cells were activated or in a resting state, when the proviruses were transcriptionally active, it was these very, very low levels of expression. So it's not the levels that were different in these either. So I'm going to summarize the findings so far, and then I'm going to tell you about a specific case. So what we found looking in the resting and activated cells of patients or donors on long-term ART is that most of these cells are latently infected. It suggests that there may be a selection for the persistence of infected cells with proviruses that are resistant to reactivation by cellular activation. So cellular activation marker expression, HLA-DR, is not a reliable indicator of proviral reactivation in donors on long-term ART. So this was a surprise to us. We really did think that these small fraction of cells within these expanded cell clones that have proviruses that are transcriptionally active would be activated cells. And what we found is that is not the case, at least after many years of ART. So we did find this very interesting case that I want to tell you about for the last few minutes here. So within these 8 donors, one thing we did is we just counted the number of infected cells within the total PBMCs, the number of infected cells within the activated subset and within the resting subset. And in the PBMCs in these individuals, it was what we are used to seeing, and that is that there's anywhere from about 10 infected cells per million PBMCs up to about 100 or so infected cells per million PBMC during viral suppression on ART. But when we looked in the resting and activated subsets, we found something interesting in one of the participants, and this participant is shown in red. So although this participant only had about 20 infected cells per million PBMCs, when we look in the activated cells, there were 9,000 infected cells per million DR positive cell. So the number of infected cells in this activated compartment was very, very high in this one individual compared to the other 8 donors. And despite the fact that there are very few infected cells when you count the total PBMCs, there actually were a lot of infected cells in this DR compartment. So we thought, well, maybe this happens to be an expanded clone that was, activated by its cognate antigen and underwent massive clonal proliferation. So how can we determine that? Well, we can do the integration site assay on this particular sample. So we collected the DR positive cells from this individual, and we performed the integration site assay. And sure enough, we found, oh, and by the way, we also did find, before I show you the results from the integration site assay, we did find that this participant had an influenza diagnosis 30 days before the sample was collected. So it's possible that this massive expansion in the DR positive subset was in response to an influenza antigen. So we performed the integration site assay to see if this is a large expanded clone. And when we did this, we found that almost every single integration site was identical in the DR positive cells. So it is one very, very highly expanded cell clone, infected cell clone. The integration site was in the MALAT1 gene. MALAT1 is a highly expressed long non-coding RNA, so it's just one big exon. It's actually a strongly preferred target for HIV integration, and the provirus was in the same orientation as the gene. So this provirus is perfectly oriented and placed to easily be reactivated if activation of these cells is enough to reactivate these proviruses, because it's in an exon of a highly expressed gene. It's in the same orientation of the gene. So this is exactly what you want to see for a provirus to very easily become reactivated. So we thought, okay, this is a perfect case where we can look at this one very, very highly expanded clone and ask and look in these activated cells and say, do they have HIV RNA with this massive clonal expansion and activation? But we first just wanted to sequence the provirus and make sure that it had everything that's needed to be expressed. So we found that although this provirus did have a large deletion in GAG and part of POL, the full LTR was there with the promoter, and both of the exons of TAT were present. So it does have what is needed to be expressed. So then we isolated single infected cells from within this specific clone. We assayed the single infected cells for the presence of unspliced HIV RNA, and we found that only 13% of them had detectable levels of unspliced HIV RNA, suggesting that most of these cells are latently infected, even when these cells meet their cognate antigen, undergo massive clonal expansion and activation. It was not enough to wake up the proviruses in these cells that persist for many years on ART. We did the same in the resting cells. We just wanted to see in the resting cells, do we detect unspliced RNA for this specific clone or not? And in the resting cells, we found that 8% of them had HIV RNA. So there wasn't a difference. So whether the cells are resting or activated in vivo on long-term ART, even if the cells meet their cognate antigen, undergo massive clonal expansion, it does not appear that this is enough to reactivate these latent proviruses within these expanded cell clones. So I'm going to come back full circle now in my last few minutes and come right back to this AMB1 clone, because I told you in the beginning, we wanted to know in this AMB1 clone, it's a very highly expanded clone with a replication competent provirus in this individual. 2% of the cells in this clone had HIV RNA, 98% of the cells were latently infected. So are these 2% activated cells? So we came full circle right back to this clone, assayed single cells from within this expanded cell clone for the presence of HIV RNA. And we did this after sorting them into activated cells, so cells that have the HLA-DR surface marker, resting cells which lack the HLA-DR surface marker, and we then determined the fraction of them that have HIV RNA. We found that 7% of the activated cells from within this clone had HIV RNA, and 7% of the resting cells from within this clone had HIV RNA. So this 2% of cells that have HIV RNA are not specifically activated cells. This 2% is comprised of both acting and resting cells that have HIV RNA, and in this case are the sole source of persistent viremia in this individual. So this was a surprising result, but it led us to this model. So from what we've learned here, we generated a model where during treatment, individuals have T cells, infected T cells that either lack the HLA-DR surface marker or have the HLA-DR surface marker. These cells, there's very low HIV RNA expression in some of these cells. If one of these cells meets its cognate antigen, as Francesco Simonetti and Lily Cohen and others have shown, they can undergo massive clonal expansion despite the fact that they're infected. What they didn't look at was whether or not this massive clonal expansion reactivated the proviruses within them, which is what we set out to do. So during pre-ART and short-term ART, which we have not yet looked at, it's possible that that is exactly what happens. So in pre-ART, it's possible that when these cells meet their cognate antigen, undergo massive clonal expansion, that it does reactivate those proviruses. And maybe even in short-term ART, it reactivates those proviruses. But on long-term ART, these populations where the proviruses were readily reactivated, easily reactivated, may have already died off. And you're left now with populations of clonally expanded cells that even when they undergo massive clonal expansion after meeting their cognate antigen, there is no change in the HIV RNA expression. So we're left with long-term ART with these populations of clonally expanded cells that even activation of these cells is not enough to reactivate the proviruses in vivo. So it leaves us with the question of if proviral expression is not associated with cellular activation on long-term ART, is it associated with integration site, epigenetics, TAT expression, or something else? And so that still remains to be found or to be determined. And of course, now we're going to go back and look at individuals in pre-therapy and in short-term ART to see if the activated cells in these individuals do have, a high fraction of them have unspliced RNA and higher levels of HIV RNA. And so the implications of this research is that cellular activation may not be enough to reactivate latent proviruses on long-term ART. The question then remains is whether latency reversing agents or other approaches are going to be any better than cellular activation. And if they're not, then maybe we should place more focus on lock and block approaches. So if 90% of these cells are already latently infected, the proviruses are not transcriptionally active, and it's a small fraction that are transcriptionally active that we know are leading to persistent viremia and probably rebound viremia, then maybe we can do the opposite and focus on these lock and block approaches, which of course many people are already doing. And so all I have left are the acknowledgements, which I sort of already did in the beginning, but of course there are many people outside of my lab who contribute to these studies by sharing samples and consulting with us. And I want to thank everybody on this slide for that. And of course, the participants for contributing their samples for these studies. And that's all I have. So I'm happy to take questions.
Thanks so much, Mary. That was really great. Are you able to see the Q&A?
I can stop sharing here.
Or I can, the first question was from Michael Luella. Michael, do you want to ask? I think there might be a way to raise your hand. There's a bunch of questions. But he asks, isn't there a third possible pathway to persistence, one that is antigen driven, responding to a pathogen or other trigger? And I think you've already talked about that in the talk, but if you wanted to expand on that, on the relative importance of that.
Yes, because I think this question was probably put in before I got to the part about the activated cells. And so obviously Michael's thinking along the same lines we were exactly. And asking the same question that we asked. Yes.
Michael Emmerman has a question. Does finding only 1 to 2 copies of unspliced RNA per cell suggest that there's quite a bit of stochastic low-level provirus activation that's independent of further antigen recognition? Did I understand your conclusion?
Yes, I think so. So, the question remains, when we detect these very low levels of unspliced RNA, how many of them are read-throughs and how many of them are actually being driven by the promoter, the HIV promoter? And so that question still remains. And I do think some of these could just be read-throughs. But then when we sequence the plasma virus, we do find that the persistent viremia matches the proviruses that are transcriptionally active in the cells when we do this assay. So, so some of these clearly are leading to persistent viremia, but so there must be maybe a very short window of time where some of these cells where we're detecting 1 or 2 copies of unspliced RNA, there might be a short window of time where they actually produce much higher levels, generate virus particles, but then die very quickly. So by the time, so when we sample, we only really can catch them in this phase where they're sort of, gearing up to produce higher levels, right? So I think that may be what we're seeing is those proviruses that have just been reactivated are just starting to make unspliced RNA. And there's a very small window of time maybe before they're dead and we can't sample them at that point.
Next question is Florian's. Florian, are you able to?
Yeah, can you hear me?
Yeah.
Yes, I can hear you.
So much for this talk, Dr. Kearney. That was super interesting.
Oh, I could hear you, but now I can't hear you.
Can you hear me now?
No, you sound very far away now.
It's very soft, Florian.
Can you hear
hear me, right?
Yep, now I can hear you.
I don't know.
Yep, better.
Okay, that's a different microphone. Outstanding talk, really interesting data, Dr. Kearney. Thank you so much. I'm just curious, how do you envision what happens during rebound when ART stops? Because the burst size for individual cells reactivating during initial infection, during rebound is calculated to be in the hundreds to thousands. And during rebound, after many years of ART, these cells would still be the ones that had been selected for low reactivation before, right? So how do you explain that during ART, the burst size per cell is so low, and then when ART stops, it becomes really high, yet ART has no effect by itself on viral reactivation?
Yeah, I actually think my guess is that they're not the same cells. So we have this low level persistent viremia. My guess is that when you start ART, those viruses that are already there can now infect new cells. And those newly infected cells are the ones that have then proviruses that are transcribed to these very high levels and lead to rebound viremia. That's my guess.
Okay, thank you.
Yeah.
Is there any other question? Oh, Marta has a question.
Oh, hi, Marta.
Marta, do you want to, I unmuted you if you want to ask it.
I can see it. So
If not, I'll read her question. She's speaking.
Sorry, can you hear me now?
Yeah.
yes, I can hear you.
All right. Sorry, I have the radio one too. It was a really nice talk. I enjoyed it, as always, with your work. I might have missed it. You may have said it and it slipped my mind. But did you detect the clones during ART longitudinally, like over many years or
Yeah.
time frames?
Yes. So in this study where in these 8 patients, we actually didn't look longitudinally with the exception of just one of them. But that one has already been reported, that AMB1 clone has already been reported that it persists for many years.
Right. And then
Yeah.
have you guys done anything to see in the sequence to see if there's CTL escapes or anything, or like try to find a reason, like maybe they're not getting targeted by CTLs?
Well, these are the individuals that we looked at were all treated in chronic infection. So they're of course going to have lots of CTL escape mutations. We didn't look specifically for that in these individuals, but we have tracked the emergence of CTL mutations in other studies, and you start to see these very early in infection, these CTL escape mutations. So, but I just don't know how many epitopes have to be mutated to the point where they're no longer expressed,
Right.
before you don't have any epitopes that are being expressed, right?
Yeah.
So
Yeah, that's a good point.
I'm not sure. Yeah,
Okay, thank you.
sure.
I don't see any other questions, but I had one.
Okay.
It was about, so with regards to integration sites, so if you had multiple full-length sequences that you got from this with the same integration site, how much variability do you see across the viral genome sequence for viruses from that same integration site?
Oh, that's so interesting. Nobody's ever asked me that question, and we're doing that study now.
Oh, really? Cool.
Yeah. Yes, we're doing that study.
That's awesome.
So I'll let you know when we have some data.
Yes.
It's not, yeah, it's a challenge to sequence full-length proviruses across many cells within a clone. And of course, most of them are just going to be identical, but we are curious to know if anything could happen to these proviruses as the cells, proliferate for many, many, many years. I mean, what about homologous recombination of VLTRs, generating solo LTRs or something? So we're looking at that now.
Cool. Great, thank you. And I don't know if there are any other questions. But if not, thank you again so much for your time and for this great talk. I learned a lot.
Thank you. Thank you so much for having me. I hope it was interesting to people because I showed a lot of data that was published, but I sort of wanted to put the new data in context. So I hope that it wasn't, people probably thought, oh, I've already seen this.
It was wonderful.
Okay, good. Thank you, Marta.
Okay.
Thank
See you later.
Okay.
Agree.
And just before people leave, the next seminar is going to be on Monday, November 8th. It's a different time to what we're used to. It's at 10 a.m. It's featuring Dr. Marcus Bugert from Karolinska, and it will be on resident memory, CD8 T cells, and HIV and SARS-CoV-2 infection. So please join us. Until then, have a great evening, night, Mary.
Yes.
Take
Thank
care.
you so much. Bye.
Yeah.